Hi, welcome to my Introduction to Biology presentation. So what we'll talk about today is first kind of the definition of life, then we'll go through three major theories that have shaped the history of biology and the development of biology as a science, and then we'll kind of talk more about those theories and interweave them with more information about the characteristics of life, and we'll kind of try to talk about how those theories have contributed to our understanding of the characteristics of life. So first, what is life? Actually, there is no definition of life, which is kind of strange. Biology is the study of life, so you would think that there would be a agreed upon definition for it, but actually there's really not. Instead of having a definition of life, we just have kind of different lists of characteristics of life. And we'll say that if you have every characteristic that's on that list, then you are considered alive. If you're missing one or more characteristics, then you are not considered alive. And it really gets complicated when you realize that different scientists will have different lists that they prefer. So different scientists will have kind of different ideas about which characteristics you have to have in order to be alive, and you'll see different lists in different textbooks. The list that we're using in here has 5 different characteristics on it. It is kind of a commonly used list of characteristics for life, and if you meet every one of those characteristics, you are a living thing, which would make you an organism. So the first thing on the list is cells. The cell is the basic unit of life. All organisms are made up of cells. So an organism like you or me is a multi-celled organism and has a lot of different cells in our body. But the simplest organisms would only have one cell. They're single-celled or unicellular organisms. That would be like a bacterium, has just the one cell. The next characteristic is reproduction. So organisms will always be able to make more of themselves in one way or another. And then you have information. So there's kind of 2 types of information that organisms will have. First, they have hereditary information, which is kind of the information that contains instructions on how to build that organism, how to build the different molecules that the organism has, and that information is passed on to the offspring when the organism reproduces. So because the information is passed on to offspring, that's why we call it hereditary information. And for all the organisms that we know of, that information is stored in the form of genes within DNA. Then you also have environmental information that organisms are able to perceive and respond to. So every organism must be able to perceive some type of information in its environment, meaning it has to be able to sense something about its environment in one way or another. Then it has to be able to respond to that information somehow or other. The next characteristic is energy. So every organism will have some source of energy in the environment, some way to get energy from the environment, and then it will use that energy to power the reactions inside the cell as part of its metabolism. So metabolism is actually all of the different chemical reactions that occur inside a cell. But usually when we talk about metabolism, we're talking mostly about the reactions that organisms use to get energy from their environment and to store it or to use it. Then next you have, or the final characteristic of life is evolution. So all organisms will evolve over time. So that doesn't mean that individual organisms evolve, it means that populations evolve. So if you have a group of organisms and you track them over time, you will see, if you track them long enough, that their characteristics change through evolution as their environment changes or just as more time is passing. Okay, so we're going to use those characteristics of life to kind of decide if a couple of different entities are alive. So first we have plants, and then we have viruses. So the first characteristic of life is cells. Plants do have cells, so they're meeting that one. Viruses actually do not have cells. Instead, they have a capsid. A cell is enclosed by a membrane, and it houses different structures that the cell needs for a living. So you'll have chemical reactions going inside that cell, a bunch of different chemicals, and I guess simple cells don't have very much in the way of structures actually, but they do have a little bit of organization, and complicated cells will have, like plant cells, will have really, well-developed structures inside the cell. Viruses just have capsids. A capsid is just a protein coat. And inside the capsid, you really have nothing except for the genetic information of that virus. Some viruses will have an envelope around the outside of that capsid, which is a membrane, but the capsid itself is not made of a membrane. So we're not counting that as a cell. There's not reactions going on in there. There's not really much organization. Inside of it, it's very simple. Very few chemicals are in there. Nothing much going on, and it's not enclosed by a membrane. It's enclosed by just a protein coating. So that's not a cell. So viruses are already not meeting one of our characteristics, which means we're probably going to say that they're not, not alive already. They're kind of already disqualified. But as we move through other characteristics, we'll see that they're disqualified in more ways than one. So the next characteristic is reproduction. Plants do reproduce. Most plants reproduce sexually using flowers, and they also will form seeds. So there's some plants that will reproduce asexually, just by making, basically an identical copy of themselves. And there's a lot of plants that can do both, but all plants are able to reproduce in one way or another. Viruses, on the other hand, do not reproduce themselves. If they don't have a host cell, there's just nothing happening with that virus. It doesn't reproduce, it just sits there like a particle. Once they get inside a host cell, it's actually the host cell that is reproducing the virus. So we don't call that reproduction actually, we call it replication. The host cell will make all the different parts of the virus and then assemble it together to make new viral particles. So that's not considered reproduction because it's not the virus doing it, it's actually the host cell that's doing it. Next we have information. So plants have hereditary information in the form of genes. Viruses actually also have hereditary information in the form of genes. For plants, their genes are always carried on DNA. For viruses, some of them have their genes carried on DNA, some of them have them carried on RNA, but they all have genes. So that's finally one characteristic that they are able to meet, at least halfway. Of course, the other half of information is responsiveness to environmental information, and that's where viruses are coming up short. So for plants, we actually kind of might think of them as not responding also, because we don't really see them moving and stuff, but they do respond. They just don't respond the way that animals respond to the environment. So for instance, if you watch sunflowers throughout the day, you'll notice that they are kind of turning to face the sun as the sun moves across the sky. Or if you have plants inside your house by a window, you might notice that the leaves of that plant or the branches of that plant are kind of growing towards the window to catch the light. Then if you move it to a different window or rotate it, now it's going to start growing in a different direction, again towards the light. Always towards the light. Also, plants, whenever they are breached in some way, like maybe by an insect that's eating them, they have defense systems actually that will be triggered by that. So for instance, they're actually able to detect, some plants, not all of them, but some of them are able to detect the sound of an insect or a caterpillar eating the leaves of the plant. And when they hear that sound, well, kind of hear it, when they detect the sound, they will respond by releasing defensive chemicals that will kind of deter that insect from eating more of the plant. So in each of these cases, they're perceiving information about the environment. They're detecting sounds. They detect sunlight. For viruses, it's not really the same. So all they really do is sit and do nothing, and then if their target cell, like their host cell, happens by, they can attach to it, and that's really it. Once they've attached to the host cell, they will kind of respond by entering it or injecting their contents inside of the host, and that's it. That's all they can do. So, I don't know, for me that kind of actually does look like responsiveness, like really, really, really limited, really, really basic responsiveness, but most scientists do not feel that way. So they're considered to not respond to their environment and not really perceive information in their environment. Certainly it's the case that if they don't have a host cell come by them, that's it. Like, they're done. They're just going to sit there until the end of time or until something happens to destroy them. They have to kind of, by random chance, encounter their host cell, and once they've encountered it, they can attach to it and, enter it or inject their contents into it. And after they've done that, that's the end of their responsiveness. After that, it's just the host cell doing everything for them to reproduce them or to replicate them. Next we have metabolism. So plants do have a well-developed metabolism. They do photosynthesis to get energy from the sun. So they convert that energy from the sun into sugars, and then they also have another type of metabolism called respiration, in which they will take those sugars that they made through photosynthesis and use them to make ATP, which is the normal kind of cellular energy, or I guess I should say that's how most cells store their energy in a form that's kind of ready to use for any purpose. So plants have a robust ability to get energy from the environment and use it to produce ATP. Then of course they would use that ATP to power different processes that are occurring inside the cell. For viruses, not the same. They don't have metabolism. Nothing happens inside of the virus until it binds the host cell, then it just enters the host cell, and then that's it. It's done. So if you look inside the capsid, literally nothing is happening inside of there. There is no chemical reactions occurring. Viruses do not have any source of energy that they use. Once they are inside the host cell, it does take energy to build new viral particles. All that energy is coming from the host cell. That's the host cell's metabolism that is being used to produce energy that then the host cell will use to replicate new viral particles. So the virus really has nothing to do with it. Some viruses actually will have enzymes inside the capsid, which are proteins that are able to do some type of function to catalyze a chemical reaction and make that reaction go faster. But those enzymes are not active when they're inside the capsid. So they're not making any chemical reactions speed up inside the capsid. There are no chemical reactions inside the capsid. Once the enzyme gets into the host cell, then it will use the host cell as an energy source. It will use ATP that's produced by that host cell as an energy source and catalyze some type of reaction that's usually used to undermine the defenses of the host cell or force it to produce new viral particles. But that doesn't really count as the virus's metabolism because it doesn't happen inside the virus. It only happens inside the host cell. And then finally, evolution. Actually, both plants and viruses have evolution, and that's just because they have genes. Any organism that has genes will evolve inevitably. So because of viruses not meeting a lot of these characteristics, they don't have cells, they don't reproduce, they don't respond to information in their environment, and they don't have metabolism. They're therefore not considered alive. But there actually is a little bit of disagreement about that. There are some scientists who say that evolution and possessing hereditary information and undergoing evolution, those are really the only characteristics of life that actually matter or should be defining. So they would say that because a virus has genes and because it evolves over time as those genes change over time through mutation, that means that viruses actually are alive. Most scientists don't feel that way, some do. Unquestionably though, viruses are related to life because, they got the genes. Okay, so now that we've gone over kind of what life is, or some ways to think about what life is, we'll talk about or just kind of introduce three major theories in biology, and then we'll start, talking about them some more. As we talk more about these theories, we'll learn about kind of this history of biology, more about those characteristics of life, and more about how biologists do science. So first we have cell theory, which is just the idea that the cell is the basic unit of life, and also that cells come from other cells. They don't come from, they don't come from other things in the environment. They only come from cells. So that's cell theory. Then you have the chromosome theory of inheritance, which states that hereditary information is found in genes, and genes are found on chromosomes. And a chromosome is just a large molecule of DNA. Then finally you have the theory of evolution, which states that new species can come from older species by evolution through natural selection. So we're going to go through each of those theories in turn, starting with cell theory and ending with evolution. Actually, though, before we talk about those theories, I just want to talk a little bit more about energy in organisms, because this is kind of the only characteristic of life, the only major characteristic of life that I want to talk about that is not touched on by those three theories. So first, just a little bit about energy. So first we'll just kind of go over the types of nutrition that cells need. So first, they have to have chemical energy. That is energy in the form of chemical bonds between atoms in a molecule. That is the type of energy that organisms will use to power their metabolism and all the different reactions that are occurring inside the cell. So they have to get chemical energy from one source or another. Like plants, for instance, are able to take in light energy and convert that to chemical energy. Other organisms need to take in specific molecules that have chemical energy, and then they can kind of harvest that energy. So if you're a plant, you're taking in energy in the form of light. If you're not a plant, you're probably taking in energy in the form of sugar or fat or maybe protein. That's some type of organic molecule that has chemical energy stored in its bonds, a good amount of chemical energy in its bonds. In any case, no matter what the ultimate source for that energy is, organisms will convert it to ATP. ATP, or adenosine triphosphate, is kind of the go-to molecule that organisms will use as a source of energy to power cellular processes. So sugar and fat, these are kind of molecules that can store energy in their chemical bonds. But if you wanted to actually use that energy to power a chemical reaction, you can't just use it directly in the sugar. First, you got to get it out of the sugar, put it in ATP, and then once it's in the ATP, then you can use that real easily to power any reaction in the cell that needs to be powered, almost any reaction. So that means sugar and fats, those are kind of like ways to store energy, like, gold maybe. If you have gold, you have money, but you can't just take it to the store and buy something. First, you got to convert it to a different form. You gotta turn that gold into cash. So sugar's like gold. It's how you can, there is energy there, and you're storing it, you got the energy, but if you wanted to actually use the energy, you gotta convert it to something else, like ATP. ATP is like cash. It's accepted everywhere. In addition to taking in chemical energy from their environment, organisms need also building blocks for their macromolecules, or their large molecules. So a building block is just a small molecule that you can use. If you get a lot of them, you could use it to build a big molecule. Those big molecules would be DNA, RNA, proteins, carbohydrates, or fats. Those are kind of the main large molecules that cells need. So some organisms can make their building blocks. Other organisms need to get the building block from their environment as a form of nutrition. And most organisms can kind of make some of them, but need to get others from their environment. Then finally, you have vitamins. Vitamin is also a small molecule, but instead of it being used to build proteins or other large molecules, the vitamin is actually used to help the proteins function. So most of the functions or processes that are going on in a cell are going to be carried out by a protein, one protein or another. Some of those proteins can do their jobs like just by themselves, if they also have an energy source, like ATP. And other proteins will have to have a vitamin as well. So they can only work if they also have the vitamin. So any vitamin is going to be, some molecule that a protein needs to function that you cannot make yourself. So different organisms will actually have different vitamins that they require, because again, some of these little molecules they would be able to make, and others they would not be able to make. So we talked about how all organisms will have some type of a way to bring in chemical energy from the environment and convert it to ATP. Different organisms will have different ways, to get their ATP, different energy sources in the environment. So for plants, their source is light. They take in sunlight from the environment and they will use it to produce ATP. They actually will kind of it's more like they use it to make sugar, and then they use the sugar to make ATP. But overall, they're converting sunlight to ATP. If you're an animal, which you are, animals will be taking in energy in the form of sugars or fats or proteins, so organic molecules that have a lot of energy stored in them, and they'll use those molecules to make ATP. Usually we like to use sugars for that process, but you could also use fats or proteins if you don't have sugar available. Then other types of organisms, which would be fungi, bacteria, and archaea, those are able to use lots of different things depending on which one we're talking about specifically. So they can use organic molecules just like animals can, the sugars, the fats, the proteins, but some of them can actually use weird stuff, especially the bacteria and the archaea. They can use stuff like methane or ammonia or different sulfur compounds. They could even use like minerals from rocks or iron even. So they, different bacteria and archaea can use a lot of different stuff to make their ATP. What each organism is able to use specifically just kind of depends on what species it is. Some of those bacteria and archaea, they don't need organic molecules at all, actually. They can just live literally on the surface of a rock by just leaching minerals out of that rock and using that to make ATP. Oh, by the way, you probably might not know what archaea are. Archaea are single-celled organisms that are really similar to bacteria, basically. So we used to think that archaea and bacteria are kind of the same, but once we started looking a little closer at their molecules, we see that there's actually some differences in the specific molecules that they have. Like, for instance, bacteria will use different fats for their membranes, and archaea will use, other fats for their membranes. They kind of store their DNA in a slightly different way, so there's a few differences between them on like a molecular level. So then once an organism has made its ATP, making it is actually part of its metabolism, but then the other part is like using it to power different processes in the cell. So for instance, sometimes organisms will use ATP to help them break down food molecules that they've taken in, or nutrients that they've taken in. They also will use ATP to build new molecules in the cell, so building new proteins, building new DNA, and of course, if a cell is going to divide, it kind of needs to make like more of all of its structures. It needs to kind of double its size, and then it can divide. So that process is powered by ATP as well. So ATP is really central to all of the processes in a cell that kind of let it be alive. And then the graphic there is showing a diagram of ATP. So this is ATP on the top. It's adenosine triphosphate. So this A ball here, that's the adenosine part. And then these three P's with little I's, those are the phosphates. It's PI because it's inorganic phosphate. So you have three phosphate groups attached to an adenosine, and that is an ATP. If you're going to use that ATP to get energy out of it, you would actually just break off this last phosphate group, and then that gives you energy. So what you're actually doing is releasing the energy that is contained in this bond between the last two phosphate groups on the ATP. When you break that bond, you would remove this phosphate, it just starts floating in the cell, and you get the energy out of that bond. And then you have to have some type of protein that is going to be able to harness that energy and use it for some purpose to power some reaction. What you're left with, or the other thing that you're left with, is a DP. So you still have your adenosine, and now it just has two phosphates attached to it, so we call it a DP now, or adenosine diphosphate. Then if you're going to make ATP, you start with your ADP, and you're going to need to basically reattach that last phosphate to it to get ATP. And that will not happen unless you put in energy. So you have to get energy from some source, maybe a sugar, a fat, a protein, sunlight itself. If you're a bacteria, maybe something weird like sulfur or iron. You're going to get energy from that source and use it to attach this free phosphate onto the ADP so that you get ATP again. You're just adding that last phosphate back on there. So this is just a cycle that will repeat, as a cell goes about its business. It will constantly be using its ATP, which gives it ADP, and it'll constantly be using its environmental energy source to attach that last phosphate back onto the ADP, giving it ATP back again. Okay, so now that we've talked a little bit more about energy and organisms, we'll start on those theories, beginning with cell theory. Cell theory is going to help us kind of learn more about, cells as the basic unit of life, also how cells reproduce, and also science itself and kind of how we perform science in biology, and actually in general as well. So since we're about to start talking about like science history, I just wanted to mention first that science history is real messy. It's usually presented as like kind of, a straightforward story, but when you actually start researching it, it gets real complicated real fast. So, for any scientific theory, we'll have like one person or maybe a couple of people that are kind of given credit for that theory. But if you actually research the history of the theory, actually a lot of different people will have worked on the theory, and only one or a couple of them at most will have gotten credit for it. So in the development of any theory, you're going to have at first, somebody propose the theory. But they would be proposing like a really early version of that theory, so what they propose might not be exactly the same as the theory that we learn about now, that we have now. And also, since it's really early, in general, like when they propose this early version, there's no evidence for it. There's no evidence to support it, which means like it is not a theory at that point. It's at best a hypothesis, but sometimes not even that. Sometimes it's just kind of an idea that they had, right? Because in order for something to be a theory, it has to be supported by lots of evidence that's come from lots of different experiments. So if you haven't done experiments to test it, then it's not a theory yet. At best, it could be a hypothesis. A hypothesis is kind of something that you think might be occurring, but it's testable. It's a testable statement that makes a prediction which you can test by using an experiment. So you can just, do the experiment and see like if the prediction made by your hypothesis kind of came true or not in that experiment. But a lot of the time when somebody proposes this early version of the theory, the technology to do the test won't even be there. So there's not even a way to do an experiment that can test this, idea that they've proposed. And a hypothesis must be testable to count as a hypothesis. So if there's no way to test it, that's not a hypothesis. That's just an idea that you had. So someone will have like an idea way early with no evidence to support it and no way to get evidence to support it. Everybody will think they're crazy, and time will pass. As time passes and developments occur, we get new technology, and when we have new technology, then we can do some of those experiments that we weren't able to do in the past. So at some point, we'll have the technology to actually design an experiment that could test this idea, and at that point, scientists will kind of start working with it, and it'll be an actual hypothesis. As people are doing more and more experiments on it, they will be changing this hypothesis, because it probably was not 100% accurate the way it was first stated. So, as you get new evidence about it, new information about it, you're going to be tweaking it. So a bunch of different scientists will be contributing to that work in general. Then as time passes and even more like new techniques are created, you'll get even better evidence for the hypothesis, and even more refined updates to it. And then at some point, somebody's going to do something big with it. Somebody's going to maybe get really, really good conclusive evidence for it, or they're going to make a really big change to the hypothesis that kind of brings it into line with everything that is known at that time, with all the new information and all the new technology that is available. So in general, like this person, if they're making new changes, that would be based on new information or techniques, and if they're getting like new evidence that's really good, that's using like a new technique that was just developed. So they're doing some experiment that wouldn't have been possible in the past. Because that person made such a large contribution to the development of the hypothesis, which is kind of now more like a theory because it's been tested so many times, they're getting credit with it. Credit for it. So generally at that point we would start to call it a theory because it has been tested many times already. It's been kind of revised to always be current with all the information that's available, that's come from all the different experiments and their results. And we've got some really good conclusive evidence at this point. So at that point we call it a theory, and somebody gets the credit for it. Whoever got the best evidence or made the latest, greatest change to it. And then things still aren't done yet, actually. People will continue working with that theory in general and continue to test it using new techniques, more experiments, and as more information becomes available, the theory can still change, actually. It may be, kind of revised to always be in line with the latest evidence. So that's a whole big process, and, out of all of that, it kind of gets hidden, and only one person, or at the most two people who are working together, will get credit for that theory. So then you just hear the simple story that so-and-so got this, made this theory, but that's not really true. Actually, another thing that makes the history of science and of scientific theories more complicated is the fact that sometimes different scientists who are not related to each other will have the same idea at the same time. And whenever that happens, we kind of pick one to get the credit and the other doesn't get credit. So a good example of that is the theory of evolution, actually. Charles Darwin and Alfred Russel Wallace both proposed the theory of evolution at the same time. They were working on like different parts of the world, like, on the other side of the world from each other, and they didn't really know each other. Charles Darwin already had all this evidence for the theory of evolution, and he was working on writing a book about it, but he was just kind of dragging his feet on publishing because he was thinking, that it might not go over so good. So he just wanted to make sure that his book was really perfect before he published. And then he got this letter from this guy, Alfred Russel Wallace, and Alfred Russel Wallace had just kind of had the idea for evolution. He didn't have any evidence to support it, but he kind of wanted to publish his idea anyways, and he thought that he should just run it by Darwin first because he knew that Darwin was like a respected scientist. So when Darwin got that letter, he was like, oh boy, I guess it's time to publish, huh? And he went ahead and published his book, and he got the credit because even though they both had the idea for the theory of evolution independently, only Charles Darwin had a bunch of evidence to back that up. Another thing that makes science history complicated is that sometimes, it's not just about the science itself. Sometimes it's about kind of other aspects of a scientist's life that will kind of prevent them from receiving credit for their work on some type of theory because it damages their reputation or, something like that. So scientists sometimes will kind of like have feuds with each other. And, if one of those scientists has like a stronger reputation in the scientific community than another, the guy who has a stronger reputation can maybe prevent, the underdog guy from getting full credit for his work, unfortunately. Also, sometimes a scientist will kind of move on to work on other stuff that's really looked down on by the scientific community, and that will kind of lower their reputation and interfere with them getting credit for their work. Also, in the past, not as much anymore, luckily, but in the past when science was kind of new, it was really kind of a white man's world, and so if you were a scientist but you were not a white man, that was a problem for you in terms of getting credit for your work, for sure. So kind of the most famous example of that is related to the discovery of the structure of DNA. You had multiple people in different labs all over the world kind of working on the structure of DNA, trying to figure it out. At that point, we knew, this is kind of like, in the early 1900s, we knew that DNA is the molecule of heredity, but we didn't know what it looks like or how it's structured or how that works. So people were trying to figure it out. And you had this lady scientist, Rosalind Franklin, who was using a technique called X-ray crystallography to try and solve the structure of DNA. In that technique, you have to get basically a crystal of DNA or whatever molecule you're looking at. You have to crystallize it, then you pass X-rays through it, and you basically develop a film of the image that the X-rays make when they come out the other side. So the X-rays will be kind of bouncing off different parts of the molecule as they pass through it, and it'll make a pattern on that film, and you can kind of calculate like the shape of the molecule based on what pattern the X-rays made as they pass through. That's really, really hard to do. It's a very difficult technique. Rosalind Franklin was spending all this time on it. It's really hard to crystallize things like DNA. It's not normal for DNA to form crystals, so just getting the crystals in the first place is a challenge. Then interpreting the X-ray crystallograph that you get once you've done the technique is also really hard as well, because, you know, it just kind of looks like, you know, patterns of light and dark, and you got to kind of figure out like a molecule shape based on that. It's real hard. So she had gotten a really good image of DNA using X-ray crystallography, and she was just taking her time, kind of trying to calculate literally the angle between like the bonds of all the atoms in that molecule, just one by one, calculating, you know, the angle that all the different bonds were at compared to each other, to get the structure. So she was kind of going about it in like the most slowest way, but also kind of the most accurate way. Then there's these other kind of two scientists, James Watson and Francis Crick, who are the ones who got credited for solving the structure of DNA. They knew that she was working on it, and they knew, because her boss had told them, that she had gotten a really good X-ray crystallograph of the DNA, and they wanted to see it, but she didn't want to share it with them. And kind of what happened is that her boss took it out of her desk and gave it to them without her knowing that that happened. And they just kind of looked at it, and they had an insight. They had like just a hunch about what the molecule would look like. And well, it turns out that hunch was correct. So they were able to publish based on that, just basically based on that hunch that they had, which they got, you know, by looking at her image. And she was just, you know, arriving at the answer much more slowly, and so she didn't get her credit, even though, you know, her final answer would have been based on a lot of math, not just, you know, a hunch. So it would have been, you know, much more, I guess, reliable. And a lot of people feel like she should have gotten the credit, but she didn't. But at least we remember her. Most people in her position don't get remembered at all. So, yeah, there's just another point to kind of bear in mind is that a lot of the time you'll have a scientist who's famous for one thing, but they did more than that. Most scientists will work on multiple things throughout their lives, so they probably made other discoveries that, you know, they aren't famous for that. And then finally, just to reiterate, yeah, like when you read about science history in a textbook, it presents it like a linear story, kind of a simple story. That makes sense. When you go research it, it's not so simple. It's real messy. It's real messy. And yeah, you'll learn about a lot of scientists who worked on that theory or that idea, and you know they get omitted from the textbook. And I thought I should just add in Rosalind Franklin's X-ray crystallograph of DNA. This is it. So she was trying to calculate the structure of the molecule based on this. She called this image 51, which I guess means that it was the 51st crystallograph she had taken of DNA, and it was the one that came out the best. Usually when they came out, they wouldn't have, I guess, as much focus as this. They would just look a lot more blurry. And so James and Watson, or James and Franson, sorry, James Watson and Francis Crick took a look at this, and they had an insight about the structure, and she was there trying to do it using math. So with all of that in mind, we'll talk about cell theory history, starting with a guy called Robert Hooke. So the development of cell theory is all kind of wrapped up with the development of the light microscope, which actually we don't really know who invented the light microscope. There's a few different people who said that they did, and you know, probably only one of them did. You know, it is possible that actually multiple people could have invented it, you know, kind of working independently at different times, but we don't really know who did. We know that it happened sometime between 1590 and 1620. And at first, you know, the microscope was not really used extensively in science. Robert Hooke is kind of the main guy who first started to use it. Actually, the main thing that microscopes were being used for at first was to look at cloth, at textiles, so that you could get the thread count and just see how high quality the cloth was. But Robert Hooke, started pointing his microscope at a bunch of different things and like drawing them and writing about it. And he published that in a book called Micrographia in 1665. He was basically the first person to just kind of look through the microscope at a bunch of different stuff and think about like what does that mean? The things that he's seeing, what do they mean? So one of the things that he looked at was a slice of cork, and in there he saw a bunch of little boxes. And they made him think about cells that monks would go in to kind of meditate and pray, and so he called them cells. He didn't understand what cells were, what their function was, how important they were. He just knew they were little boxes, and they reminded him of a monk's cell, and he called them cells. Well, that's a cell. So he was looking at cork cells, and yeah, he's the reason why we have the word cell, even though he didn't know like the importance of the cell at all. And that image there is a reproduction of the microscope that he used. So you can see it's very pretty and pretty simple. This is actually a drawing that he made in Micrographia of what he saw of the cork cells. So he looked at the cork slice, or actually a couple different slices that are from different angles. And in this one, he saw all those little boxes that he called cells. The next major contributor to cell theory was Antoni van Leeuwenhoek, who's actually the man responsible for discovering microbes. A microbe is any organism that's too small to be seen with the naked eye, so you have to see it by looking at it in a microscope. He actually published his discovery in 1674, so before that time no one knew that there was such thing as organisms that you can't see with the eye. He didn't call them microbes or microorganisms, he called them animalcules or beasties. And he could tell they were alive because he saw them like moving around in his microscope. He actually described all different types of microbes that we know of today, except for viruses, because viruses are even smaller than the other microbes. Since they don't have cells, the capsid is actually like crazy small for almost all viruses. There's a few that are large enough to see with a regular microscope, but most of them are way too small for that. So he wasn't able to discover viruses, but he found all the other types of microbes, even though he didn't really know like what they were or how to classify them. He at least recognized that they were alive. He actually made his own microscopes, which were really advanced for the time. The lenses that he made for his microscopes could just magnify things much larger and with much finer focus than any other lens at that time could do. But he was kind of really jealous of his inventions, so he wouldn't let anybody look through his microscopes. So if you wanted to see, what he was seeing, you had to look at his drawings. He wouldn't let you look through the microscope. Like not even the king, not even his wife got to look through the microscope. And when he died, he actually had never told anybody how to make those special lenses that he figured out how to make. So, no one else was able to make microscopes that were that advanced. And it took about 100 years for other scientists to figure out how to make lenses that were of a similar quality. So this is an example of his microscope, like a drawing of his microscope. He actually made a lot of different scopes like this, and he would just kind of glue his specimen to the scope. So if he wanted to look at something else, he had to make a whole new microscope. And these are some images he made or drew of the microbes that he saw under his microscope. Cell theory continued to be developed in the 1800s. Henry Dutrochet was one of the very first scientists to propose that the cell was actually the basic unit of life. And not only was it like a structural unit, but it was a functional unit as well. However, it's other scientists who are actually credited with cell theory. So after his proposal, you had a number of other scientists working on cell theory, including Theodor Schwann, Matthias Schleiden, Robert Remak, and Rudolf Virchow. Out of all of those people, it's actually just Theodor Schwann and Matthias Schleiden that have received credit for cell theory. Schwann stated that animals are entirely made of cells, and Schleiden stated that plants are entirely made of cells, so that like every structure within the body of a plant or an animal is just composed of cells. So I'm not sure exactly, to be honest with you, why it is that their contributions were considered more significant than others so that they received credit for the theory, but that is how it went down. In the pictures here, this guy on the top is Henry Dutrece. This is Matthias Schleiden, and this is Theodor Schwann. Rudolf Virchow also made really important contributions to cell theory. In 1858, he proposed that cells come from other cells and can only come from other cells. So previous to that time, most scientists believed in spontaneous generation, which is the belief that living things come from non-living things spontaneously. So, people didn't really understand where life comes from, but they knew that, like, when you leave out a piece of meat, after a little while you get maggots on it. So they actually kind of thought that the meat makes the maggots, and that is spontaneous generation. The idea that something that is non-living, like a piece of meat, can spontaneously generate living things like maggots. Actually, this idea had been around for centuries, and most scientists who were alive at that time accepted it. But Rudolf Virchow did not accept it, and he stated that actually cells do not come from non-living things. In fact, they can only come from other cells. So this sparked a kind of a big debate in the scientific community of that time over, where it is that cells come from, where it is that living things come from, and it was in this debate that scientists kind of formalized the scientific method. So before we go on to talk about that debate, I'll just go over the steps of the scientific method, which you probably remember most of from high school. So first you start with observations. So you're kind of looking at the world around you and you're seeing some stuff. After you've made your observations about the world, they kind of cause you to form a question. So you have some kind of question about these observations, what they're really about, what can explain them, something like that. Once you have your question, you make a hypothesis. So the hypothesis is basically an answer to that question that you made. It's what you think the answer would be based on everything that you know about science, about the world, and you would actually also do some research to see what other scientists have found out that could be applicable to your question. So you would base your hypothesis on previous research as well. In order for your hypothesis to be a valid hypothesis, you have to make sure that it makes some type of prediction that you can test. So you have to have some way of figuring out, like, is this hypothesis accurate or is it not accurate? By doing an experiment, which will tell you basically if it's accurate or not. So in that experiment, you are testing your hypothesis and you're looking to see what happens as the outcome of the experiment. If the outcome matches with the predictions of your hypothesis, then you could accept your hypothesis. If the outcome does not match, then you could reject your hypothesis. But first, we have our experiment with control groups so that we can make sure our experiment is working correctly and have something to compare our data against to see, how to interpret it. After you do the experiment, you make observations based on the experiment. So that's basically collecting your data from the experiment. Once you have those observations, then you're going to interpret them. And after you have interpreted them, you can reach a conclusion, either that your data support your hypothesis and they match the predictions made by your hypothesis, or that your data do not support the hypothesis and do not match the predictions made by your hypothesis. So if your experimental data do not support the hypothesis, you have a couple of options. First, you could just reject that hypothesis and kind of start over from scratch. Second, you could modify that hypothesis. So just change it a little bit, kind of tweak it to be more in line with the actual data that you got from your experiment. Then after you've modified the hypothesis, you would do that experiment again using the modified hypothesis and get your new observations and see whether or not they support your modified hypothesis. So if your data do support your hypothesis, then you get to accept your hypothesis. So at that point, probably what you're going to do is publish. But in terms of the overall, project of science, after you've accepted the hypothesis, your next step or somebody's next step would be to repeat the experiment. So you have to do the experiment again, and actually, ideally, you would do a different experiment that can test the same hypothesis in a different way or using some different techniques. You would want to kind of repeat those experiments over and over again. You can repeat the same experiment multiple times just to make sure it comes out the same way every time. You can also do a different experiment that's testing the hypothesis in a different way, just to make sure you have multiple kind of lines of evidence that are all supporting that hypothesis. Once the hypothesis has been tested a lot of different times, probably by a lot of different scientists working in different labs and using different techniques, then you can say maybe that your hypothesis has become a theory or even a law. So if it's a theory, that means that it has been supported by reams and reams of evidence. Lots of experiments have been done on this hypothesis and it was supported every time. So we're really, really certain that it's correct. Of course, since we're in science, we're never going to say that we're 100% certain that it is correct. We'll never say that it's just like completely proven. We always kind of want to leave the door open for us to be wrong about something, for there to be something that we didn't know about yet. So that's why we say it's a theory and not like, a universal truth or something. A law, on the other hand, is just something that is observed the same way over and over and over again. A theory is more like an explanation of something, and a law is just the observation that happens that way over and over and over again. So for instance, there's a law of gravity which states that when you have an object held and you let it go, it will fall. It happens every single time. But that doesn't explain like why that is happening. Then there's a theory of gravity that tries to explain why that's going on, well, what's really happening there. And that theory would be saying that it's about like the mass of the Earth and stuff like that. So that's the scientific method. Now back to spontaneous generation. So actually, spontaneous generation had already been disproved for organisms that you can see with your eye a long time ago by the mid-1800s. So actually, a scientist called Francesco Redi had disproved it in 1668 by doing an experiment where he had these kind of beakers that he put meat in. He had one beaker, which was his control, that he just put some meat in it and he left it open to the air. He had another beaker that was an experimental treatment where he put the meat in the beaker and then he sealed it with some cork. And he had another experimental treatment where he put the meat in the beaker and sealed it using gauze. And then he waited to see what would happen. So in his control beaker, he noticed that flies came in and you got maggots on the meat. On the sealed beaker that was sealed with cork, there were no maggots that developed on the meat. In the beaker that he had sealed using gauze, he noticed that flies came and they would kind of chill out on the gauze, and then there ended up being maggots on the gauze, but none on the meat. So he figured that what's actually going on here is not that the meat is making the maggots, it's that the flies are making the maggots. The flies are attracted to the meat, and then they're trying to get on that meat and lay some eggs that will hatch into maggots. He didn't realize that there were eggs, or how exactly they were creating the maggots, but it was pretty clear that they were creating the maggots somehow, and it certainly wasn't the meat. So actually in the gauze-covered beaker, the flies could kind of smell the meat through the gauze, and that's why they came to hang out on the gauze and lay eggs there. So this was a really important experiment because it proved that spontaneous generation was not correct for visible life forms. It was also important because he used a control condition. He used that unsealed flask. So he was one of the first scientists who actually used controls in an experiment. And controls are really important in an experiment because they let you see that your experiment is working correctly, and they give you something to compare your data to help you interpret it better. So he had his flask unsealed with the meat in there, and he got maggots on there. But what if there ended up not being any maggots on that meat? Well, then he would know that something's wrong with his experiment. His experiment doesn't allow for the potential detection of maggots forming on meat, even if that would happen under the best circumstances possible. So if he got no maggots forming on that meat in the unsealed flask, he would know that he needs to change something about his experiment. In order to prove that maggots don't form on meat if it's closed off to the air, you have to be able to conduct that experiment under conditions where it would be possible for maggots to form on the meat if that was something that was going to happen. So even though Francesco Redi had disproved spontaneous generation for visible life forms, once microorganisms were discovered, scientists still said, like, could be true for microbes though, could be true for beasties, you don't know. So Leeuwenhoek had actually, gotten some raindrops and washed them under his microscope, and he noticed that they kind of started out clear, and then as time went by, they got cloudier and cloudier, and he saw more and more little animalcules inside of them, and he was like, well, where are they coming from? It's like they're coming from the rain. So he kind of believed in spontaneous generation for his little beasties. You also had another scientist, John Needham, who had done an experiment where he took some beef broth and boiled it, and then he put it into flasks and corked them to seal them. And then he just kind of watched what happened to it, and he noticed that the beef broth got cloudy over time. It was getting cloudy because microbes were growing inside of it. So that experiment was actually done in the 1740s, and because of it, people kind of thought that, microbes actually do come from, non-living things like beef broth spontaneously by spontaneous generation. So the debate over spontaneous generation kind of continued after that time, and there was another scientist, Lazzaro Spallanzani, who did another really important experiment that kind of contradicted what John Needham's experiment had shown. So he was thinking that there was actually some problems with John Needham's procedures, because what Needham had done is, like, he boiled his beef broth, but he didn't boil it for very long. So Needham was thinking, or I should say Spallanzani was thinking, that there could have been some microbes maybe that survived the boiling process, because it just didn't last for very long. And then what else Needham had done was just like let his flasks sit with the beef broth in them before he sealed them. He actually let those flasks cool down back to room temperature before he sealed them with corks. So then it could have been possible for microbes in the air to fall into the flask during the time that it was cooling and before it got sealed. So Lazzaro Spallanzani thought that maybe those are the reasons why John Needham had gotten the results that he did in his experiment. And he did another experiment that was published in 1799, which he basically kind of repeated John Needham's experiment, but put a few twists onto it. So he boiled his beef broth for a lot longer, and then he sealed it right away. He didn't let it cool down. And instead of sealing it with cork, he actually kind of melted the necks of his flasks and sealed the glass. So this is kind of a diagram showing Spallanzani's experiment. So he had his control condition is on the top, and his experimental treatment is on the bottom. In both cases, you have a flask full of beef broth that he boils for quite a long time to make sure everything in there is dead for sure. Then the control flask, he just left it open, And the experimental treatment flask, he sealed it right away by melting the glass of the neck closed. So this flask did not cool down before he sealed it. And then he just waited to see what would happen. And he noticed that in the control flask that was open to the air, it got cloudy and it filled up with microbes. On the experimental flask where he had sealed it, the beef broth remained clear and microbe-free. So he concluded that actually spontaneous generation does not apply to microbes, and beef broth does not create microbes. Actually, what had been happening is that microbes are in the air, they fell into the beef broth, and they ate it and made more of themselves. Other scientists at the time, though, didn't really accept his experiment. They started criticizing his methods. They said that, like, in those flasks that he had sealed super good by melting the glass, there actually wasn't enough air left in there for anything to live no matter what. And they said that he boiled the flask for too long. So I guess they think that kind of like he boiled it for so long that he altered the structure of the beef broth maybe so that it couldn't support life. So they were saying that basically in his experimental treatment, there's no way it could have supported life anyways. So of course he didn't see microbes growing in that beef broth. So this debate continued until finally in 1861, Louis Pasteur fixed it for everybody by doing the ultimate experiment that no one could find problems with that disproved spontaneous generation at last for microbes. So he also was kind of tweaking previous experiments. He kind of took Spallanzani's experiment and tweaked it. So he also had beef broth in a flask, and he boiled it. He didn't boil it for quite as long as Spallanzani, just to make, the detractors happy. And then he didn't seal it by melting the flask completely shut. Instead, he kind of heated the neck of that flask, and then he pulled it into an S shape. So Louis Pasteur's experimental setup is kind of on the bottom here. So he had these flasks with beef broth, and he heated that broth, and then he heated the neck of the flask and pulled the glass to make an S. So these flasks are open to the air. Air can get in, so no one can say that there's not enough air for life. There's plenty of air. But if anything's kind of falling in, any little particles or microbes in the air, if they're kind of falling into this flask, they're just going to fall right here into this S, into the bottom of this S. There's no way they're going to be able to climb this glass tube and enter the actual flask with the beef broth. So he made his flask like this, and then he just kind of waited, and he saw that his beef broth remained clear and microbe-free actually for 18 months. He left some of these flasks for 18 months, and he didn't see any microbial growth. Then at the end of that period, he actually took some of these flasks and tilted them so that the beef broth made it all the way into this little bend in the S, and then he put them back upright, and he kept watching them, and he saw that at that point they actually did get cloudy and had microbes growing in them. So he was able to really conclusively, conclude that, the beef broth does not create microbes. That doesn't happen. Actually, microbes are in the air. They fall into things such as beef broth and then start eating that broth and grow and flourish. So he was able to kind of prove that there had been microbes floating in the air, or at least, something floating in the air that fell into the S of that flask and was able to, reproduce in the beef broth when it was actually exposed to the beef broth. And this satisfied all the other scientists at the time. And they all said, well, I guess spontaneous generation's wrong. So that was a debate on spontaneous generation. And here's kind of our modern conception of cell theory. So our modern understanding of cell theory is not just that the cell is the basic unit of life and all life forms are made of cells. It's also that cells come from other cells. So spontaneous generation is not correct, and cells can only come from other living things, other cells. So we say that cells divide, they split into two to make, two new cells. So that means that for all organisms, for all multicellular organisms, all of the cells in that organism's body have originally come will have come from one original cell that divided over and over and over again to form all the cells in the body, which would include your body, of course. So you start with a fertilized egg, which is also called a zygote. It's just an egg cell that fused with a sperm to fertilize it. That is your initial cell. Every other cell in your body comes from that initial cell. So this is kind of an image of your initial cell. Actually, you can see this little crescent in the back. That's the second cell. So when this first cell divides, it makes that second cell in the back. Then all the cells start dividing. So both of these two cells will divide to make 2 more cells. Then all 4 of these cells will divide to make 8 cells. And this process will continue until you form like a ball of cells with a bunch of cells. And then it starts, making structures. And, from there you eventually get, an embryo and a fetus and then a person. So in addition to our understanding that cells only come from other cells, we have the understanding that for all the cells in your body, or for any multicelled organism, all of the cells in that organism's body all contain the exact same DNA, except for any mutations that may have occurred, during the developmental process. So, because it's one cell with, its set of DNA dividing over and over and over again to make all the other cells, all those cells are going to get that same DNA, unless there's some mutations that occur. So that is kind of interesting because we don't have cells that are all the same, right? Like our cells look lots of different ways. So we have muscle cells that are all long and spindly. We have blood cells, red blood cells that are like little packets that don't even have a nucleus in them. We've got nerve cells that send little branches off like trees in all different directions. We've got cardiac muscle cells in the heart that are long and spindly and branch. We've got kind of square-like cells in the liver, and we've got in the intestine or lining the intestine also little square-like cells, but they got little hairs on the top, kind of, or little like little tubbies, like little nubbins on the top. All of these types of cells have come from the stem cells that form, basically that form an embryo. So they all have the same DNA because they're all from the same place originally, and yet they're all so different from each other. So how could it be that they're all so different when they have the same hereditary information in their DNA? And the answer is because within that DNA, you've got, all the genes for all the types of cells, and each type of cell is only going to use the genes that it needs for that cell type. So the nerve cell uses the genes for nerve cells. It doesn't use blood cell genes. The muscle cell uses genes that are for muscle cells. It doesn't use liver cell genes. The cardiac muscle cell uses the cardiac muscle cell genes. The skeletal muscle cell doesn't use genes that are specific to cardiac muscle cells. That gives us all the different cell types in our body. There's only kind of one big question that's left by cell theory, which is, if cells can only come from other cells, then how did we get the first cell? Like, where does the first cell come from if there's no cell for it to have come from? And so we don't exactly know the answer to that question. This is something that would have happened billions of years in the past, which makes it really difficult to figure out what had gone on. But our best idea so far is this idea called chemical evolution. So whether chemical evolution is a theory or a hypothesis kind of depends on who you're talking to, but most scientists accept that this is like probably the way that it happened, and we're just kind of working out the details of how it would have occurred. But in the idea of chemical evolution, we're saying that we have inorganic molecules existing probably in the oceans of the early Earth that combined together, that reacted with each other using energy that comes from lightning or from deep sea hydrothermal vents to drive those reactions. And they react together to form organic molecules. First, they form simple organic molecules, and then later on, those simple molecules react together to form complex organic molecules. And at some point, those complex organic molecules kind of develop into a protocell or an early not super a life form, but like a proto-life form. So scientists think that this could have occurred kind of probably in the deep sea vents, and maybe you would have had cycles where some of these chemicals kind of evaporated into the air, went up into the atmosphere, and then were hit by lightning, which drove reactions, and then they fell back into the ocean in the rain and sank down back to the deep sea vents. And, you know, there's a lot of research to be done in this area, a lot to still be worked out, but this is so far our best idea on where the first cell would have come from. So next we'll talk about the chromosome theory of inheritance, which is going to help us learn more about how hereditary information is kind of stored and transmitted in organisms, which is of course one of the characteristics of living things, having hereditary information. So the chromosome theory of inheritance states that hereditary information is, located inside of cells on large molecules called chromosomes, and that on those chromosomes the information is arranged into units that are called genes. So hereditary information is any information that would be passed on from parents to offspring that is going to include information for the different traits that that can be passed to parents, well, from parents to offspring. A gene is a unit of hereditary information, and in general, every gene codes for one protein, which means that the gene has the instructions for how to build one protein. So every protein would have its own gene that has the instructions on how to build it. And then the proteins are going to form most of the structures in the cell and do most of the work within the cell. Either they'll be forming things directly or they will be building other things that will, form structures in the cell. So a chromosome is basically just a large molecule of DNA. So you have in every cell a number of these really big molecules of DNA called chromosomes, and then the different parts of them are the genes. So this theory was first proposed in 1902 by Walter Sutton and Theodore Boveri. The only kind of caveat is that at that time, no one knew what DNA was. So we knew that hereditary information is passed on from parents to offspring, but we didn't know, what molecule is responsible for that. We didn't know that DNA exists. So there was actually a search for, what is the molecule of heredity? And then after we eventually discovered that it was DNA, then there was a search for, what is the structure of DNA? How can we explain, like, how it kind of works? So in 1902, when this theory was first proposed, of course it wouldn't have been a theory at that point yet. It would have been more like a hypothesis or even just a good idea. They just kind of had this idea that, there would be molecules or kind of large units of hereditary information called chromosomes, and that if you looked at those molecules, you would find smaller units within them called the genes that had specific instructions for different proteins or different parts of the cell. So this image here is showing a diagram of a chromosome. This is kind of the typical chromosome structure that we're familiar with. So we normally think of it as being an X, and so this kind of swiggled up blue line is the actual DNA double helix that's just packed really tight to form the chromosome. Actually, chromosomes will only look like this when the cell is about to divide. At other times, they are not this tightly packed, so they more are forming just kind of a mass, and it doesn't really look like anything so special. It doesn't have like a structure that you can just kind of see with your eyes. But when the chromosome is condensed like this, you can kind of map where different genes are on the chromosome, and this is an example of such a map. So actually each half of this X is identical. So even if you have just one half of the X here, that could be a chromosome too if it's not attached to the other half. And then to get the X, you would take this one half and copy it, and that gives you the other half of the X. So of course you need 2 copies in order for the cell to divide, so that each new cell can get 1 copy. So if you had just 1 copy, that would look like this, like a line like this. And this kind of constricted part is this part right here, where the 2 lines of the X would like cross each other. So these kind of black bars here are showing the locations of different genes on this particular chromosome. And some of them are kind of labeled with the name of the protein that that gene codes for. So for instance, right here, this black line right here, or this black bar right here, that is the gene for EPO, or erythropoietin. Erythropoietin is actually a hormone that we need to produce red blood cells. So the kidneys will release EPO as a hormone whenever you need more red blood cells to be made, and it'll go to the bone marrow and cause the stem cells there to form more red blood cells. So if you don't have this hormone, you actually can't really make new red blood cells, which is a problem. And I guess another interesting thing about EPO is that it is kind of a drug of abuse for some athletes. Since if you kind of take it like as a drug, then you're going to end up producing more red blood cells, and then, you know, you'll have more ability to carry oxygen in your blood, which just kind of increases your endurance for sports. So once we kind of did figure out that DNA is the molecule of inheritance, we still didn't know what the structure of DNA is, or like how it could operate as the molecule of inheritance. So it was actually in the mid-1950s, in 1953, that James Watson and Francis Crick proposed their model of what DNA might be structured like. And I guess I kind of went over this story before, but yeah, they jacked Rosalind Franklin's X-ray image, X-ray crystallograph, which is again, you know, shown over here. You know, they didn't tell her. Her boss took it out of her desk. She had no idea. And, you know, once they looked at it, they kind of, you know, got a flash of insight about what the structure might be. And they realized how, like, if DNA had this structure, it would be easy to copy the information and pass it on to offspring. So they thought they were probably, you know, maybe right about that structure. And they published based on it. But in that publication, they actually didn't make any mention of, you know, her X-ray crystallograph being the one that they used. So they kind of acted like she didn't contribute to their insight at all, which is even, you know, even a bit worse than just stealing the image. They stole the image and then they just took the credit. But, you know, anyways, the idea that they had was that DNA is a double helix. Which means that it has these two strands that are kind of wound around each other. That is the basic shape of a double helix. And then they said that within, or like, I guess between the two strands of the double helix, you would have these other parts of the molecule stretching across, which are called bases, and they would form pairs with each other. So that it ends up looking like a ladder, and these paired bases are the rungs of the ladder, and then the ladder is just kind of twisted up to form the helix. So they also propose that there are 4 different types of bases that you could have: A, T, C, and G. A is adenine, T is thymine, G is guanine, and C is cytosine, and that they can only pair in certain ways with each other. So C can only pair with G, and A can only pair with T. And they realize that if that's the structure of DNA, then you can kind of easily copy it without making mistakes in the information. So those bases end up being, you know, part of those are actually the information. It's the order of the bases in that ladder of the double helix that is containing the information, or the hereditary information. So each base is sort of like a letter in a word. And then you could conceive of the whole word being like, you know, the gene. Yeah, so that's a pretty long word actually, but something like that. So this diagram is kind of showing, you know, the insight they had about how hereditary information could be passed on if it was, or if DNA was structured in the way that they proposed. So if you have this double helix with 2 strands, and you have these bases forming pairs with each other that make like rungs of the ladder, and if there's only so many bases and the bases can only pair with each other in certain ways, then if you were to separate these strands, you would be able to rebuild new strands using each one as a template. So first you've got this original DNA molecule, and then down here you're separating it, kind of pulling the 2 strands apart. So this is actually what happens whenever you are replicating or copying DNA. The 2 strands get pulled apart. And then you've got these bases, which each of these bases would be like 1/2 of the rung of the ladder. It needs another base paired with it to be the full rung. But you can, because you know the rules about how they pair, you can figure out what should go there. So if this is an A, T pairs with A, so that should be a T there. This is a G, G pairs with C, so that has to be a C that goes there. This is a T, that pairs with A, so you need an A to go there. Another T, so you need another A there. And you can just build the whole new strand. You can build a whole new strand using each of these original strands as a template without making any mistakes, theoretically. So in that way, you can kind of like copy the information over, and you would end up with 2 new molecules of DNA, not just the one that you started with. And if you had cells that were going to divide, you could put 1 in each cell, and they would all have, you know, the same information. Actually, there are sometimes mistakes made when you are copying DNA in that way. So sometimes you might have a T, so you need to put an A, but actually the enzyme that's actually doing this kind of messes up and it puts maybe a C instead. So whenever that happens, that's actually called a mutation. Anytime you have a mistake in the genetic code, that's a mutation. So normally it's kind of important to not be having those because, you know, you need to get the instructions right so that you'll have genes that can build the right proteins. But actually in the long run for like life as a whole, we actually do need those mutations because that's the ultimate source of the diversity of life. If DNA was always perfectly copied every single time, then there would be no evolution. There would only be, you know, whichever single-celled organism was the first organism, that's all that there would be still. So we wouldn't exist at all without mutations. Even though if we get a mutation, that might be kind of bad for us. You know, if they didn't exist in general, we wouldn't exist in the first place, so I guess that would be bad for us too. Once you have a mutation in there, cells will actually have ways to try and fix it, but if they're not able to fix it, then it's going to be it'll become permanent actually. That mutation will then be copied over into new molecules of DNA as though it was just kind of the way that it's supposed to be, so that all cells that are descended from that cell that has the mutation from that point on, they're also going to have the same mutation. So that was kind of how DNA is copied, just like a really brief overview of how DNA is copied. But DNA isn't just copied, it's also used to make proteins, or the genes within DNA are used to make proteins. The central dogma was originally stated by Francis Crick in 1958, and it basically describes the way that information flows from DNA to proteins. So the original idea was, you know, proposed by Francis Crick in 1958, but it was revised later on by James Watson in 1965. When they had this idea, there actually wasn't any experimental evidence for it. So it was just kind of an idea that they had. They also didn't really have like a way to test it at that time. But since then, we found that it's pretty right. And I guess at first we thought that it was like super right, all the way right, and that's why, you know, it's the central dogma. But actually, as it turns out, it's only pretty right. It's right most of the times, but there are some exceptions. So anyways, the central dogma just says that DNA makes RNA, RNA makes protein, and proteins do work. So you start with your hereditary information stored in DNA. Then if you were going to use that DNA to make protein, first you would actually make an RNA copy of the gene, then you would use that RNA copy to build the protein, and then the protein does the work. And then we have names for each of these processes. So DNA being copied into RNA is transcription, RNA being copied into protein or being used to make protein is translation, and then of course when DNA is copied to make more DNA, that's replication. So as I kind of mentioned, this is like basically right. There are some exceptions to this kind of scheme of, you know, information flow. For the big exception, I guess, is that certain viruses can actually, they will have their genes stored as RNA and they will copy them backwards into DNA. Like HIV, which is the virus that causes AIDS, will do that. It has RNA genes and it copies them into DNA and then it puts them in your cells. Well, you know, Yeah, it can't do that unless it's already in your cell, but it will put that DNA copy into your own DNA actually. But in general, that is accurate. It goes from DNA to RNA to protein, and then proteins do work. So let me go into more detail on some of these terms. So first, DNA. We've probably all heard of DNA. DNA actually stands for deoxyribonucleic acid, which is kind of a fancy word. Its structure is the double helix with the base pairs, and its function is to store hereditary information for organisms and to transmit it to offspring. When you have DNA in organisms, it's going to be always kind of organized into these large molecules that we call chromosomes. And then if you look on each chromosome, you will see segments of the DNA that are arranged into units called genes, and every gene codes for a protein. Again, there's a couple of exceptions to that, but in general, every gene codes for one protein. And for sure, every protein has a gene that has the instructions for how to build that protein. But if you were to actually build the protein, first you have to take the DNA and put it into the form of RNA. So you would find the DNA like in that specific gene for the protein that you're trying to make, and then you're going to make an RNA copy of that gene. So RNA is another molecule that is very similar to DNA, but a little bit different. It's not deoxyribonucleic acid, it's just ribonucleic acid. Actually, the difference between them is just one oxygen atom. So deoxy means, without that oxygen. So ribonucleic acid has an extra oxygen. Deoxyribonucleic acid does not have that extra oxygen. Even though chemically that's like the only difference between them, the structure of them is also kind of different. DNA has two strands that form a double helix. RNA almost always will just have one strand that also has bases on it, but not base pairs, just one base. At each rung of the ladder, it's like a half rung. You have basically a half ladder with RNA. RNA is not used to store information. It actually has a number of different roles in the cell. One of its main roles is to just carry information in a gene from DNA, out of the nucleus so it can be used to make protein. The type of RNA that does that is called mRNA or messenger RNA. So the other functions that RNA can have, at least some of them we'll talk about later on in the semester. And then finally you have proteins, which are really big molecules that have a really complicated 3D shape. And they're used, as I had mentioned, both to build structures in the cell and to carry out different functions in the cell. So here's some figures kind of showing those structural differences. Here you have DNA, the double helix. Here you have RNA. You can see it looks really similar, but it's only one half of the ladder. It only has one strand. Instead of having base pairs, it just has single bases. Other than that, it's pretty similar. So you would have information in DNA in a gene, and you're going to copy that information into RNA in a process called transcription. And then once you have your RNA molecule, or specifically your mRNA molecule, you can use that to build a protein through a process called translation. And here is an example of a protein. So proteins have all different types of ways that they can look, actually. Every single protein has a unique 3D shape. So they don't all look like this one. Some of them look very, very different from this one. But this is just an example. So this type of protein is actually an antibody that would be used by your immune system to target a specific pathogen and destroy it. So here you have kind of a figure showing where this would be occurring in space in the cell. So your DNA is of course stored within the nucleus of the cell. If you're going to make proteins only get made in the cytoplasm of the cell, which is the space outside the nucleus. So you have to get this information from the DNA out of the nucleus before you can build a protein. And that's kind of the reason why we have mRNA. So first you would find your gene on the DNA, then you're going to do transcription on that gene and create a molecule of mRNA based on that gene. It has all the same information that that gene had. Then that mRNA molecule will leave the cell and find a big protein complex called a ribosome. And the ribosome is going to read the mRNA, and it actually will be able to kind of tell what protein it should be building based on the instructions in this mRNA, which came from the DNA. So this process of a ribosome using mRNA to build a protein is called translation. So here I have just kind of an analogy that you could use to understand the central dogma. So in the central dogma, you start with DNA. Information in DNA travels into RNA. Then that information in RNA travels into protein, and proteins do work. If you were working with music, you would start with sheet music, which is kind of storing the information about the piece of music to be played. If you were going to actually play that music, then you might make a bunch of copies of the sheet music and distribute them to members of an orchestra. And each member in the orchestra might put their own annotations on the sheet music to make it kind of their own. So that sheet music used by people in an orchestra would be corresponding to RNA. Then they're going to use that sheet music to make music. And RNA would be used to make protein. Then proteins do work, and music, when you hear it, well, it creates like art and enjoyment. So there, the art and enjoyment are kind of corresponding to the work in a cell. I also have an analogy to kind of computers. So you have, information stored in DNA. In a computer, you have your information stored on the hard drive of the computer. So the hard drive would be like DNA. Then if you were going to work with that information, you can't just use it directly from the hard drive. You actually have to copy it into the RAM, which is like the working memory of the computer. So the RAM would actually correspond to RNA in a cell. Once you have your RAM, you can use that, or I guess the computer would use that, to display a user interface for a program that you are using. That user interface could kind of correspond to a protein in a cell. And then, proteins do work, and you are going to do some work using that user interface. You're going to, get something done. So here with the exceptions, so there are a few exceptions to the central dogma. So the central dogma states that information can only flow from DNA to RNA to protein, and then proteins do work. We actually have some exceptions there. So first you have reverse transcription, where certain viruses can start with RNA and go back to DNA. You actually have some RNA that's able to do work. Not a lot, but even actually in the ribosome is probably where you have the most important example. There's actually some RNA within the ribosome that doesn't just carry information, it's doing work. It actually does some of the work of building the protein. So those are exceptions to the central dogma. You also have a couple of others. You have actually a number of others. They're kind of rare. But for instance, you have proteins that can kind of be used to modify stored information in DNA. That's in a process called epigenetics. Epigenetics is information that is passed on from parents to offspring. Actually, specifically, it's only from a mother to her offspring, but this information is not stored in the DNA itself in terms of like the sequence of bases in the DNA. Actually, it's stored in proteins that are attached to the DNA. So for instance, if you have a woman who is like severely malnourished as a child, that's going to cause certain changes in her DNA. It's going to cause certain proteins to attach to her DNA. So it won't cause a change in the actual like sequence of DNA. It doesn't cause a mutation, but it makes certain proteins attached to the DNA. Then let's say that her circumstances improve and she gets access to plenty of food. So she goes on to have like a nice life. But she's actually probably going to never reach her full height because when she was a child, she didn't have enough nutrition to grow to that full height. If later on she has children, those children actually are going to not grow to their full height that would be encoded by their DNA. They're going to be short as well, shorter than their DNA would say, like for them to be. And that's actually because of those proteins that are attached to the DNA that were passed on to them from their mother. Basically, the idea is that It's kind of a way for organisms to adapt to their environment more quickly than you can do through like regular mutations and evolution. So, if the mother was exposed to these really harsh circumstances where there just wasn't enough food, then maybe those circumstances might affect the offspring as well. And then it would be good if they're not trying to grow like higher or taller than what there is like nutrients to support. That's basically, the idea. Then you also have prions, which I'm not sure if you guys have heard of prions, but you've probably heard of mad cow disease, which is a disease that's caused by prions. So a prion is actually just a protein that is misfolded, so it has the wrong shape, basically. It has the wrong shape, which means that it can't do its job. And not only that, but if you put it around other proteins that are the same type, it'll make them misfold the same way so that they get the wrong shape too, and they stop working too. So if you get some of these prion proteins, for instance, by eating like infected cow meat, they will start making your proteins change their shape to be the wrong shape. Your proteins will stop working, at least that type of protein will stop working, and this kind of just progresses. It happens, throughout your life. These diseases take like decades to develop, but eventually you're going to end up with these really large masses of proteins in your brain that are not shaped right, and they start killing your neurons, and you die at the end. So that's almost like the information in the misfolded protein being transmitted to other proteins nearby it. So that's like a type of information flow as well. So those are all exceptions to the central dogma. So it might have occurred to you that it's kind of weird for the central dogma to be called dogma. A dogma is a belief that cannot be questioned. So in science, we question everything. We never consider anything to be fully proven, like for all time. We always, we value questions. So it's kind of weird that we have this idea in biology that's called a dogma. And the reason why why it's called a dogma is actually because Francis Crick decided to call it that, and he didn't know what the word dogma meant. So he has said, like as a quote, I guess later on he wrote this quote, which is just, as it turned out, the use of the word dogma caused almost more trouble than it was worth. Many years later, Jacques Monod pointed out to me that I did not appear to understand the correct use of the word dogma, which is a belief that cannot be doubted. I used the word the way I myself thought about it, not as most of the world does, and simply applied it to a grand hypothesis that, however plausible, had little direct experimental support. So that's why he called it a dogma, and now we're all just kind of, stuck calling it a dogma. But it's not actually a dogma. You're free to question it. And actually, we have discovered exceptions to it, by questioning it. So it's not an absolute kind of rule. And just, to reiterate, in science, we do not have dogmas. So the last major theory that we'll talk about today or in this chapter, is the theory of evolution. Which we're just going to kind of go over pretty broadly, and then we will come back to it later on in the semester, actually at the very end of the semester. So the theory of evolution was first proposed in 1858 by Charles Darwin in his book On the Origin of Species. Actually in the same year, it was also proposed by another scientist, Alfred Russel Wallace, who was working basically on the other side of the world from Charles Darwin. And I guess I kind of went over this already, but so Charles Darwin had been gathering evidence for the theory of evolution for a long time. So he had already had this idea, years before 1858, and he had gathered a bunch of evidence to support it during his travels around the world, and he was kind of compiling everything that he had together into his book, but he was worried about publishing it because he knew that it wouldn't go over too well, and that he would get a lot of pushback on it. And so in 1858, this guy, Alfred Russel Wallace, had also had the idea for evolution, but he didn't have evidence for it. And he wrote to Darwin, just kind of asking him for his advice on whether he should publish it. And so Charles Darwin said, wrote back and said, yeah, you should publish it. But at the same time, he was like, yeah, I need to publish now as well. So he went ahead and published his book. And so even though both of them proposed a theory at the same time, of course, at that time, it wouldn't have been considered a theory, more like a hypothesis. Charles Darwin is the one who gets credit for the theory of evolution because he had a book full of evidence, whereas Alfred Russel Wallace didn't have any evidence at all. Since then, evolution has been supported over and over and over again in experiment after experiment after experiment, in observation after observation after observation. So there's a lot of evidence for it, and it is solidly a theory. The theory of evolution states basically that all species are related to each other by common descent, which means that they all share a common ancestor with each other, and that species are able to change through time and sometimes form new species, and that this process of change in species over time is driven by natural selection. So I guess as we go through this, we'll be defining a few terms. So first off, the term evolution. Evolution is a change in the characteristics of a population over time, which is important. I guess the part there that's really important to note is the fact that it's in a population. So a change in the characteristics of an individual is not evolution. Only a change in the characteristics of a population would be considered evolution. Then a population can be defined as individuals that all belong to the same species that are living together in the same place at the same time. So natural selection is the main process that drives evolution. So we will define what that is and kind of go into that a bit. Natural selection is this process in which traits that are advantageous are more likely to be passed down to offspring than traits that are not advantageous or traits that are disadvantageous. So if you're an individual who has a trait that is advantageous, you are more likely to pass that trait on to your offspring than another individual who has traits that are disadvantageous, which means that if you're looking at the offspring, more of them will have the advantageous traits and less of them will have the disadvantageous traits. So over time, the advantageous traits are becoming more and more common in the population, which means that the population's characteristics are changing and the population is evolving. So natural selection will actually occur inevitably if you have several different assumptions that are met. So first, you have to have different individuals within the same population that have different traits and different characteristics. Second, you need those traits and characteristics to be heritable, which means they can be passed down to offspring, which means that they are genetically determined traits. So they're not based on things that happen to you in your life, they're based on your genetics. And then third, you need to, the last assumption that needs to hold true is that some traits are kind of more helpful for surviving and/or reproducing, and other traits are less helpful for surviving and/or reproducing. So if those three things are true, you have individuals that have different traits in a population, their traits can be passed down to offspring, and some of those traits are more helpful for surviving and having more offspring than others, then if you're meeting those three assumptions, then natural selection will inevitably occur. It's impossible to avoid it. Basically, if you have an advantageous trait that makes you better at surviving, or maybe it makes you better at finding a mate, something like that, then you're going to have probably more kids. If it's a trait that makes you better at surviving, then you're just, you're going to live longer, and in a longer lifetime, you're going to have more opportunities to have children. You're probably going to have more children overall. Then your children are all going to get those advantageous traits that you had, and they themselves are also going to be benefited and end up having more children of their own, so that over time, those traits are becoming more and more common in the whole population. On the other hand, if you have a disadvantageous trait that makes it difficult for you to survive, or maybe it makes it difficult for you to have a mate or find a mate or something like that, you're probably going to have less children. So if you're living less long, then you're just, you have fewer opportunities to have children, you're just going to have a shorter life. That probably ends up meaning that you have less children. Those children that you do have actually will all have that disadvantageous trait that you have, which means that they also will not have a lot of children. So over time, the number of individuals in the population that have that disadvantageous trait is going to go down and down and down. So natural selection is actually acting on individuals. An individual will have advantageous traits or disadvantageous traits. An individual will have more children or less children. But evolution as a whole, sorry guys, that was my cat. Evolution as a whole acts on populations. So an individual either has children or doesn't have children, or has more children or has less children. But the proportion of individuals in the population that have a certain trait changing over time, that's what evolution is. The figure there is kind of an example, a common example of natural selection and its effects on a population. So on the right-hand side, you have a tree that has white bark, and you've got some moths hanging out on that tree bark. So this is actually something that was observed over time in forests in England. So there was a particular forest in England that has trees that used to have a white bark like this, and moths that lived in that forest would be mostly white, and they kind of blend in really nicely with the tree when they're white. You would have some moths that had a mutation that made it so that they were dark in color. So they don't blend in at all. They really stick out. So in this situation, when the bark of the tree is white, then it's beneficial if you're a white moth. That's an advantageous trait, being white, because it lets you blend in with the tree, and it makes it hard for predators to see you. So when this bird comes to try to eat some moths, it's going to be really easy for the bird to see the dark moths, and it's going to eat a lot of dark moths. It's hard for the bird to see the white moths, so not as many white moths are going to end up being eaten. That means that over time, dark moths are being eaten more, they live less long, which probably means they have less children. These white moths aren't eaten as much, which means they live a long time, they probably have a lot of children. So as time passes, you're going to see less and less of these dark moths in the population. And indeed, you would see kind of very few of them in the population. But what happened over time is that eventually the Industrial Revolution occurred, and cities started producing a lot of pollution from factories, and all of that pollution in the air ended up landing on the tree trunks and made them turn dark. So in the same forest, many years later, you actually had trees that had a dark bark because of all of the pollution. And so now the situation for these moths is very different. Now it's good if you're a dark moth. That's become an advantageous trait because the environment changed, and now the dark moths can blend in with the tree trunk, and the white moths are really sticking out like a sore thumb. So now when the bird comes to eat some moths, it sees the white moths really easily, it eats them more, and the dark moths are kind of hidden, so the bird has a hard time finding them. So now, over time, what's going to happen is that these white moths that used to be favored, they're not favored anymore. They're being eaten more quickly, they have shorter lives, they have fewer offspring. These dark moths that used to have a really hard time, now they have an easy time. It's hard for them to be eaten, and they live a long time, they have a lot of children. So over time, the population of moths in the forest is going to evolve. As more and more dark moths have children and less white moths have children, you end up having a lot of dark moths in the population. So we had a population of moths that started out being mostly white, and when the environment changed and the selective pressures changed, that population of moths evolved by natural selection to be mostly dark. So a few more terms in evolution. First, we have speciation. Speciation is when a new species is created from a previous species. So anytime one species kind of evolves, or a population that belongs to that species evolves to form a new species, that is what we call a speciation event. Then we have fitness. Fitness is specifically, it's an individual's ability to produce viable offspring compared to the same ability of other individuals in the population. So that means not just how good are you at surviving, how good are you at finding food, that also means how good are you at finding a mate, how good are you at having children, and even how good are you at raising your children. So, your children, in order for you to be fit, to have a really high fitness in evolutionary terms, you have to have a lot of children, and not only that, your children need to have children of their own. That's what I guess that's what we mean when we say that the offspring are viable. We mean that they're able to have children of their own. So, For instance, if you have, just tons and tons of children, but, as soon as you have a kid, you eat the kid, that means that you are not fit. Your offspring are not viable because you're killing them. So it doesn't matter how many you have in evolutionary terms, you're not passing on any of your traits in a way that impacts the population. So in order for you to be fit, not only do you have to survive, but you have to have kids, and those kids need to go on, they need to grow up healthy and strong and have kids of their own. Technically speaking, if you die after you had your kids, then you're still equally fit. It doesn't matter. So this is not the same thing as survival. Usually when we think about fitness, we think about how good are you at surviving or how good are you at competing for resources. It actually doesn't mean that at all. Survival can kind of impact your ability to have children, but it's not the same, of course. It's of course not the same thing as your ability to have children. So really what matters for fitness is how many kids do you have and how many of them go on to have kids of their own. Then finally we have adaptation. Adaptation is any trait that is heritable, so you can pass it on to your offspring, and it increases the fitness of individuals who possess it in their current environment. So when the trees are white, then being a white moth, or the trait of white color in moths is an adaptation. But when the trees turn dark in their color, now the dark colored moths are the ones that have the adaptation. So the adaptation is any trait that you can pass on and helps you survive in the environment, or helps you have children in your environment, which helps you be more fit in that environment. The image here is actually several different finches. So finches are one of the kind of main types of animals that Darwin studied and wrote about on the origin of species. So he had taken a voyage to the Galapagos Islands, and on all the different islands he noticed that they had different finches. And they're all pretty similar to each other because they're all finches, but when you look at the beaks, then you can really start to see some big differences between these finches. So these are all actually different species of finches. What happened is that you had a finch, or like a couple of finches, come over from the mainland and end up on one of those islands in the Galapagos. And they, hung out there. They had a lot of kids. They populated the island, and they adapted to that island. And then you started to have members of that population leave the island and go to other islands in the Galapagos. So every island in the Galapagos ended up having its own kind of species of finch, because everywhere that these finches showed up, they ended up adapting to their environment through natural selection, and they ended up speciating to form new species of finches that had traits that were kind of individually suited to the conditions on their particular island. So because evolution tells us that, or lets us know that all species are related to each other by common descent and we all share a common ancestor, that means it should be possible for us to draw a tree of life, which is kind of similar to like a family tree, but it includes all organisms on the planet. All organisms theoretically that have ever existed would be on the tree of life. So we're working on this. We don't got it done yet, but we're working on it. Especially at kind of the base of the tree where we're looking at the origin of life, it gets a little bit difficult because all of those species are now dead. But at the very base of the tree of life, you have an organism called Hooke. Hooke is the last universal common ancestor. So that is the last organism from which all of us are descended. And when I say all of us, I mean all species that exist on the earth today. So that includes us, humans, all of the other animals, but also all of the plants and all of the bacteria and all of the archaea and all of the fungi. Every single species on the planet is descended ultimately from Hooke. That is the last ancestor, or the most recent ancestor, that all of us share. So Hooke is not exactly the first organism to exist, but Hooke is really close to that first organism that existed. At least that's what we believe. And of course, we don't know what species Hooke was, or we would give it a species name. So Hooke has not been identified, but we just know that it had to have existed. So this tree of life that we're talking about is a type of phylogenetic tree. A phylogenetic tree is, a diagram similar to a family tree that is representing evolutionary relationships between organisms. And phylogenetics is the field of science in which you study evolutionary relationships between organisms. So what we're trying to do is figure out, which species descended from which other species and how recently and what all those relationships are, and then we can map them all out onto a tree, a phylogenetic tree. There's actually, this diagram over here is a phylogenetic tree. This is not the actual tree of life. It doesn't have all life forms on here. In particular, it doesn't have bacteria or archaea, but this is essentially all eukaryotic life. So all organisms that have a nucleus in their cells would be eukaryotes. They're all basically on this tree. So at the bottom of the tree here, we have protists, which are kind of single-celled organisms that have a nucleus, and then things start branching off. So the plants diverged a long time ago, and then you have kind of the animals. And fungi would be on here somewhere as well, but they're not, well, they're actually not shown on this tree, are they? So it's apparently not a complete tree. But so here we are, and you can see that we are closely related to gorillas, or other large primates, other apes specifically. Our little branches off from theirs, really close, not far away, so we're really closely related to them. Birds, on the other hand, we're a little bit more distantly related to birds, and you can see that the birds actually evolved from the reptiles. So that's an example of a phylogenetic tree, not a complete one, of course. It's actually pretty hard to build phylogenetic trees. It's kind of hard to know how closely organisms are related to each other, or what exactly the evolutionary relationships between organisms are. Usually we would try to figure that out by looking at genetic sequences. So we would be looking at the DNA of different organisms. Sometimes we're looking at their RNA. Usually we would look at their DNA though. And just comparing how close those sequences are. So if we're looking at the pattern of A's, T's, C's, and G's, how close is that pattern in different species? If it's really close, then we figure that those species are closely related to each other. If it's not very close, then we figure that they're not very closely related to each other. And that's basically how we're trying to figure out the relationships and kind of build a correct tree of life. So of course, as I had mentioned, the tree of life is not done, but we are always kind of working on it and adding to it, and it does get better and better and better as time passes. So far in our tree of life, we've identified three different domains of life. So there's three kind of really major groupings of species that are all, sharing some similarity to each other. And this is actually, on the side here, a diagram that's actually the tree of life. This is not a complete tree, because, there's a lot of life. So there's a lot of little entries missing from this tree. But it is at least summarizing the whole of life. So we have the eukaryotes on here, we have the archaea, and we have the bacteria. Those are actually the three different domains of life. So the bacteria, as you can see, that's really the biggest domain by kind of a lot. Bacteria are all single-celled organisms, and they are prokaryotes, which means that they do not have a nucleus. Archaea are also single-celled organisms that are prokaryotes, so again, no nucleus, but they're actually different from bacteria. If you start looking at them on a molecular level, you see that they are not the same. How they store their DNA is different. Some other things about them are different as well. Their genetic sequences are different. A lot of the time you also find them living in extreme conditions. You also have bacteria that can live in extreme conditions, and by extreme I mean in places that are really hot or really salty, stuff like that. But it's just, bacteria live kind of everywhere, including extreme places. Archaea also live everywhere, including extreme places, but when you look at the extreme places, it's they've got a higher proportion of archaea than other places have. And then finally you have eukarya, the domain eukarya, also called eukaryota, which would be the eukaryotes. Most of them are multicellular. Some of them are single-celled though. All of them are eukaryotes, which means that they have a nucleus, and inside the nucleus they store their DNA. So you've got here at the very end of the tree you have Animalia, so that's, we're on there, all the other animals are on there with us. You also have fungi there. Then you have the plants. See, the plants are actually here in Glaucophyta. And the rest of these groupings are mostly single-celled eukaryotes with a few multicelled in them. So maybe I actually misspoke when I said that most eukaryotes are multicelled. They're probably actually mostly single-celled as well, but most of the ones that we think about are multicelled. Yeah. And then, of course, at the very bottom, you have Hooke, the last universal common ancestor. And all the other branches of the tree of life kind of branch off from Hooke. So one thing that you can see when you look at the tree of life here is that archaea are more closely related to eukaryotes than bacteria are. So bacteria branch off from archaea right after Hooke lived, and then the eukaryotes actually branch off from archaea right here. So that's a ways after Hooke had lived, and yeah, we are actually more closely related to archaea than to bacteria. And that's borne out in our genetic sequences and also in some of our characteristics. For instance, the way that we store our DNA actually looks kind of similar to the way that archaea store their DNA. So in addition to phylogenetics, which is the study of evolutionary relationships between organisms, we also have taxonomy, which is the science of naming and classifying organisms. So that's really closely related to phylogenetics, but it's actually older than phylogenetics. We had a taxonomy a long time before we really understood like what DNA is or how traits are passed down, or the fact that evolution happens, or how organisms are related to each other. So we kind of made this initial taxonomic framework for classifying organisms and naming them, and now we're having to kind of update that to bring it in line with the relationships between organisms that we're learning about in phylogeny. So within taxonomy, a taxon is any group of species that are grouped together as being similar and that has a name at any level in the taxonomy. So you'll usually have different levels, like you would start actually with a domain, and then beneath that you might have a kingdom, and then phyla, so on and so forth until you get to genera and species at the bottom of the taxonomy. So each of those levels are levels in the hierarchy where you have taxons. So, for instance, bacteria, the domain name, that's a taxon. Eukarya, that's a taxon. Taxonomy is important because it helps us kind of try to wrap our heads around the diversity of life, and also because it helps us communicate about different species and different groups of species without confusion, even if we are working in different parts of the world than other scientists, or even if we have different languages and different cultures from other scientists. So this is, ideally, this is one standardized system that is used by all scientists working all around the world to communicate with each other very clearly about what they're talking about, which is actually really important. So when I was doing my dissertation research, I was actually in a neuroanatomy lab, which means that I was studying the structure of the brain. And one of the things that I was doing was kind of comparing different maps that we have, or that different neuroscientists have created of the brain. Each one is called an atlas, and it just shows like the different structures in the brain and where they're at. So there's actually a few different ones, and I was trying to figure out like how they can compare to each other and how we can, bring data from one into another. And it was really, really difficult because the makers of these different atlases did not agree on what structures there were there or what they should be named. So for me, it was really difficult to figure out if a structure with one name in one atlas was the same thing as a structure with a different name in a different atlas. And when you're looking at, the papers, people are using whatever naming system they kind of, were trained in, and it's not necessarily the one that you were trained with, and it's really hard to kind of get your mind around exactly what they're talking about when they're using a different naming system than the one that you are familiar with. Yeah, it's very difficult. So neuroanatomy would really benefit from like a shared taxonomy or something like that, where we're just using one standardized naming system for structures. That would be really, really nice. It would have saved me many hours of headache. So, maybe one day, but in any case, taxonomy is really important. The first kind of major taxonomist was Carolus Linnaeus. I'm sorry, you guys, that was my cat again. In 1753, he published the first taxonomy that was trying to include all life forms, so all of the animals and all of the plants. He actually didn't really know about other life forms at that point, so he didn't include them in his taxonomy. He actually included minerals as well in his taxonomy, and he was actually just basing it on kind of shared characteristics. So organisms that looked like other organisms, he would put them together in a taxon. Sometimes those organisms are actually not related to each other at all, though, which means that his taxonomy kind of needed to be changed like a lot when we figured out, how genetics works and, what the evolutionary relationships between organisms actually are. But his taxonomy is still important today because we're still using his kind of naming system. Like, he's the one who came up with the terms of kingdom and phyla and stuff like that. So we're still using that. He's also the first scientist to use the word species, and of course we still use that one. He also came up with something called binomial nomenclature, which is the system used to name any species. Basically, in binomial nomenclature, you put the genus name and then the species name, with the genus name capitalized and both of them in italics. So for us, our species name is sapiens and our genus is Homo, so we are Homo sapiens. So that system for naming species is still used today. Over here you have a page from his taxonomy, and then up at the top you have a couple of different plants that he actually classed together in a taxon because, as you can see, they look really, really similar, but they're actually not closely related at all. So when we start looking at their genetic sequences, we see that they're really different and they're not closely related. They should not go together in a taxon. So phylogeny is really causing us to have to make a lot of changes to the taxonomy that he created. This is actually an example of convergent evolution, where you have two different species that are not closely related to each other, but they're exposed to the same kind of pressures in their environment because they live in a similar environment. And so they end up kind of evolving similar traits or similar adaptations to help them live in that environment. So for these two plants, they are living on completely different parts of the world, but they're both living in deserts, and they've evolved this shape to help them kind of survive the harshness of the desert through convergent evolution. So in modern taxonomies today, we are still using Linnaeus's hierarchy. We're still using the way that he named species. We did add one thing to his hierarchy. We added domains. So in Linnaeus's hierarchy, the highest level was the kingdom level, and now we have domains. The domains were actually added pretty recently in the 70s, in 1976, by the scientists Carl Woese and George Fox. They're also the scientists who discovered archaea, and when they were analyzing the sequences, specifically the ribosomal sequences for different organisms, for bacteria, for these new archaea that they discovered, and for eukaryotes, they figured out that they're different, and they all kind of fall into like three different groups, or one of three different groups. So there's one group that's for bacteria, one for archaea, and one for eukaryotes, and that's why they made the three different domains. So apart from keeping Linnaeus's hierarchy, except for adding domains, and keeping his naming scheme for species, we've pretty much changed all of the other things because he was just classing things based on what, if they look similar, and we're trying to class things based on how closely they're related to each other through phylogenetics. So that means that we've had to change a lot of stuff. When we're trying to do that, when we're trying to look at genetic relationships and use that to suss out the evolutionary relationships between organisms, we like to choose genes that are what we call highly conserved, which means they don't change very much over time because they're really important and you need them. So, for instance, let's see, I guess the sodium-potassium pump, which is it's a protein that we're going to learn about later in the semester, but every cell has it. It's really, really, really important. You really need it. So that's what we call highly conserved, which means the gene for that protein doesn't change very much, because when you get a mutation in that gene, probably what happens is that the protein doesn't work right. It doesn't pump the sodium and the potassium correctly, and then you die. So for any protein where, like, if it doesn't work right, you're probably going to die, that means that that gene is probably highly conserved. It's not going to change very much because every single change that happens, for the most part, it results in you dying. And then you don't have children and you don't pass that trait on. So for these really crucial genes that are highly conserved, they're not changing much over time, which means that even if we're trying to look at vast stretches of time, like millions and millions of years, we can still track that gene and see that it's changing, not that much over time. And that really helps us look at the relationships between things. If we have two species with basically an identical version of that gene, then we know that they're super close together. If it's a little bit different, we know that they're pretty close together, and probably it'll never be very different because, they would be dead, but it just helps us track things changing through time when things are changing very slowly. For other genes that are not as crucial, they can actually be changing quite a lot over time, and then it gets hard to track things if you're looking at a large swath of time. So this slide is titled Modern Taxonomies, plural, because there's actually more than one. Different scientists have kind of different ideas about what a taxonomy should look like. And, especially for organisms like bacteria, we really don't know that much about them in a way. We know a lot about certain species, but most species of bacteria that are out there, we really don't know much about them whatsoever. And that makes it really hard for us to kind of figure out how they would be related to each other. Also, there's multiple genes that you could be looking at. It's not just one that you rely on. So then maybe for a couple different species of bacteria, one of those genes that you're looking at is pretty close between them, but another one is pretty different. And then you kind of have to make like a judgment call about how closely related they are to each other. So it's definitely not an exact science, and there's a lot of different kind of opinions or viewpoints out there on how organisms are related to each other exactly, which means that we have multiple different taxonomies. We have not multiple different naming systems. We have like kind of international bodies that are in charge of keeping us like a coherent and consistent naming system that we will all abide by. But in terms of how things are related to each other, there are different ideas about that. Finally, on the side here, we have kind of the hierarchy of taxons that we use in most modern taxonomies. So at the highest level, we have just life itself. And then within life, we have three different domains, bacteria, archaea, and eukarya. And then it kind of changes a bit depending on which domain you're talking about. But for eukarya, after domains, you have kingdoms, then you have phyla, then you have classes, then you have orders and families and genera, and finally species. If you're talking about bacteria, they actually skip the kingdom level and go straight to phyla.